Today we discuss human health and diseases, particularly NCERT exemplar problems and typical problem regarding this chapter. So first one is read the statements and find the correct ones. So incorrect and correct statements only typical, difficult to answer. So you should read the statement carefully first. IgE antibodies are produced in an allergic reaction, correct only. If any person is suffering from allergy, IgE antibodies are produced more in number. By simple blood test, the doctor identify whether that person is suffering from allergy or hypersensitivity with the help of blood test. So that is first statement correct. Second, B lymphocytes mediate cell-mediated immunity, wrong. T lymphocytes mediate cell-mediated immunity. So what is the function of B lymphocytes? B lymphocytes produce antibodies. Antibodies produced by plasma cells, one of the types of B lymphocytes involved in humoral immunity. So second statement wrong. Third, yellow fluid colostrum, abundant IgE antibodies, wrong. Abundant antibodies in colostrum are IgA type. So third statement wrong. Fourth, spleen is a secondary lymphoid organ. So 1 and 4 correct. So what is a secondary lymphoid organ? The lymphoid organ in which mature lymphocytes or immunocompetent cells become functional, known as secondary lymphoid organ. Another definition, the lymphoid organ provide antigen for immunocompetent cells, also known as secondary lymphoid organ. Similarly, the lymphoid organ provide place for interaction of immunocompetent cells and antigen, also known as secondary lymphoid organ. So answer is D, 1 and 4 correct. Next question, match the column. Natural active immunity. Natural active immunity is due to natural infection. So A3. Next one, natural passive immunity. Natural passive immunity, so from mother, transferred from mother. So A3 and B is 4. Next one, artificial active immunity. Artificial active immunity is due to vaccination. So next one, artificial passive immunity, anti-rabies serum. So what is natural active immunity? Natural active immunity means if pathogen enter. What is active immunity? If pathogen enter in our body, immune system produce antibodies, that is active immunity. What is natural active immunity? So during day-to-day activity, some pathogens enter. At the time antibodies are developed. That is natural active immunity. So while suffering from natural infection, we develop immunity. That is natural active immunity. That's why, so answer is natural active immunity acquired after smallpox infection. So natural infection. Next one, natural passive immunity. So what is passive immunity? Inoculation of preformed antibodies. Antibodies is known as passive immunity. Preformed means already. Mother transfer without her notice, transfer antibodies to fetus, transfer antibodies to infant through colostrum. That is natural passive. Next one, artificial active immunity. Intentionally we inoculate pathogens to prevent dreadful diseases. So that is vaccination. Artificial passive immunity, anti-tetanus serum injection or anti-venin injection regarding snake bite. Example for artificial passive immunity. So answer is 3. Next question. The cell-mediated immunity inside the human body is carried out by T lymphocytes. So T lymphocytes and natural killer cells. Here option only T lymphocytes. B lymphocytes, humoral immunity. Thrombocytes, auxiliary cells, help in immunity. Erythrocytes, cellular barriers. Cellular barrier. So answer is T lymphocytes. So T lymphocytes, cell mediated immunity, particularly TC cells and natural killer cells. Active TC cells also known as killer cells. Killer cells and natural killer cells involved in cell mediated immunity. So fourth one, match the following column. Natural active, artificial passive. So different natural. So natural active fighting infection naturally. So A4. Next one is artificial passive. Artificial passive is injection of anti-tetanus serum. Natural active is fighting infection 4. Artificial passive is 3. So second option. Similarly, artificial active induced by vaccination. Natural passive from mother to fetus. Next question. Which one of the following statement is correct with respect to immunity? Preformed antibodies need to be injected to treat the bite by a viper snake. So anti-venom injection. Viper is a venomous snake. Correct. So why? Whenever a snake bites, our immune system not respond. Snake venom, our immune system not consider snake venom as antigen, so no response. So for the survival of snake victim, we inject the already formed antibodies, that is known as anti-venom injection. Second, the antibodies against smallpox pathogen are produced by T lymphocytes. Wrong. T lymphocytes never produce antibodies. Third, antibodies are protein molecules. Correct. Each of which has 4 light chains. Wrong. 2 heavy chains and 2 light chains. Rejection of a kidney graft is the function of B lymphocytes. So it is also wrong. Rejection by T lymphocytes. T lymphocytes involved in organ transplantation rejection reactions. So answer is A. Next question. Study the following sentences. The cells of malignant tumor divide erratically. So divide continuously. Correct only. They are malignant tumors of epithelial cells. Actually, you should read which of the above are true for angiosarcoma. What is angio? Angio means blood vessel. Sarcoma. Cancer to connective tissue is known as sarcoma. But here given second option, epithelial cells. Cancer to epithelial cells is known as carcinoma. That's why second statement wrong. Next one, they are malignant tumors of organs that originated from mesoderm. Correct. What is sarcoma? Cancer to any tissue developed from mesoderm known as sarcoma. So third statement also correct. Fourth one, these tumors are found in organs such as spleen and lymph nodes. Spleen and lymph nodes are lymphatic organs. So first one correct and third one. So answer is C. So lymphoma, not sarcoma. Cancer to spleen and lymph nodes. So that is known as lymphoma, not sarcoma. That's why first statement and third statement are correct. Next question. If you suspect major deficiency of antibodies in a person, to which of the following would you look for confirmatory evidence? How can you confirm in that person antibodies are very low? So that is serum globulins. Globulins present in the blood. So alpha, beta, gamma globulins. Gamma globulins are antibodies. So by counting the serum globulin, so they came to know the antibodies are deficiency or sufficient number of antibodies present in the body. So serum globulins. Immunoglobulin, antibody is known as immunoglobulin. Immunoglobulin is one type of globulin. So serum contain alpha globulin, beta and gamma globulin. Some gamma globulins converted into, so they are one type of globulins, antibodies, gamma globulins. So answer is serum globulins. Autoimmunity is caused due to, what is autoimmunity? In some patient, unfortunately, immune system recognize self as a non-self. What is the main function of immune system? Identify self and non-self. Self means our own body part, non-self means foreign. Once the immune system recognize any cell, tissue, organ as a non-self, try to destroy. So in case of autoimmunity, immune system of patient recognize own cell, tissue, organ as non-self and destroy. So that is autoimmunity. Ability of immune cells to damage self cells, correct. Second, ability of immune cells to discriminate between self and non-self, no discrimination. They identify own cells as non-self. So second statement is not applicable to autoimmunity. Third, inability of immune cells to damage self cells which represent antigens. So they destroy, no inability. So this statement also not correct. Fourth one, inability of cells to distinguish self and non-self. So that is also correct. Inability. So they considered their own cells as non-self. So answer is D. 1 and 4 correct. 1 and 4 correct. Autoimmunity. Next question. The odd one out in the following is spleen and Peyer's patch. Spleen is a secondary lymphoid organ. What is Peyer's patch? Peyer's patch is the mucosa associated lymphoid tissue present in small intestine. So both are lymphatic organs. Secondary lymphatic organ. Second, spleen and thymus, both are lymphatic organs. Spleen is a secondary lymphoid organ, thymus is a primary lymphoid organ. Third, liver and spleen. Liver is not a lymphatic organ, so third one is wrong. Fourth, thymus and Peyer's patch. Thymus is a primary lymphatic organ. Peyer's patch is a secondary lymphoid organ. So answer is only 3. Only 3 is odd one. So next question. The anatomic barriers is formed by anatomy. So anatomical barriers. So physical barriers here prevent, resist the infection. Skin and cilia, mucosa, mucous membrane. Bile and enzymes. So enzymes, that is physiological barrier. Leukocytes, cellular barrier. That's why answer 1 and 2. 1 and 2, anatomical barrier. So that is physical barrier. Physical, not physiological, physical barrier, anatomical barrier is formed by 1 and 2. So leukocytes, cellular barriers. Bile and enzymes means physiological barriers. Next question. Which of the following statements are correct? IgM is the first immunoglobulin produced in a primary response to an antigen. Whenever antigen enter, first responded antibody is IgM only. Reason, IgM is pentameric, contain 10 paratopes. That's why whenever pathogen enter, first responded antibody IgM, later responded antibody IgG. In our body, 75% antibodies are IgG type of antibodies. So correct only. Second, IgD protects the body fluids and also stimulate phagocytes and complement system. Wrong. IgD stimulate the immune cells only. IgD, the antibodies present on the surface of the B lymphocytes are IgD. These antibodies stimulate. So the B lymphocytes undergo division, produce plasma cells and memory cell. Plasma cells produce and release circulating antibodies. So IgD responsible for the formation of more antibodies only. They never stimulate phagocytes and never stimulate the complementary protein. So wrong. Third, IgE act as a mediation in allergic response. Correct. 1, 3 correct. Fourth, IgE protects from inhaled and ingested pathogens. Wrong. IgE antibodies are formed in patients suffering from allergy. 1 and 3. D option correct. Next question. Physiological barrier, cellular barrier, cytokine barrier, inflammatory barrier. Physiology means by function. By function resist the infection. Here cerumen. Cerumen is secreted by ceruminous glands, wax, prevent the entry of pathogen. So A3, only one option. So answer B only. Sometime cellular barrier, cells, neutrophils engulf the phagocytic cells, engulf the antigen and destroy the antigen. So cellular barrier. 4. Next one, cytokine barrier, interferon, interleukins and interferons. ILS, IFS are known as cytokines. Interleukins secreted by immune cells. They stimulate the immune cells. As a result, they undergo multiplication. While discussing cell-mediated immunity, humoral immunity, we discuss interleukins secreted by T helper cells. In humoral immunity, stimulate the B lymphocytes. B lymphocytes undergo division. Interleukins secreted by T helper cells in cell-mediated immunity stimulate the TC cells. TC cells undergo division. Two types of cells are formed. Effector TC cells and memory TC cells. So these are interleukins. Interferons. The viral affected cells produce interferon. Interferon prevent the infection to unaffected cells. So cytokine barrier, interferon. Next one, inflammatory barrier, histamines. Histamines, bradykinin secreted by mast cells, basophils and blood platelets are inflammatory. That's why basophils, mast cells, blood platelets are known as auxiliary cells, helping cells in immune system. So answer is 2. Next question. Match the following columns. IgE antibody, allergic response A1, IgA protect developing protection of newborn babies. So protection of newborn babies. Colostrum contain abundant amount of IgA, protect the newborn. So correct. C, IgD activate B cells. So you already know, we already discussed the IgD antibodies, the surface antibodies of B lymphocytes. Surface antibodies of B lymphocytes stimulate the B lymphocyte. B lymphocytes undergo division. As a result, plasma cells and memory cells are formed. So IgD. So function is activate B cells. Next one, IgG protect developing fetus. So answer is A. Next question. Lysozyme kills bacteria by destroying their cell wall. Lysozymes present in tears, saliva. So the lysozymes destroy the cell wall the bacteria fail to survive. In this manner, bacteria dies by the action of lysozymes present in tears and saliva. Answer is cell wall. An antiviral substance produced in response to viral infection for restricting its multiplication is interferon. Antiviral protein interferon prevent viral infection to healthy surrounding cells. So 15th answer is C. The regions of an antibody that make it distinct from all other kinds. You already know 5 types of antibodies. So the variable region, the classification of antibody into 5 types depending upon the amino acid sequence in variable region. Constant region means the constant region of all 5 types of antibodies contain same amino acid sequence. Difference only in the variable region. So answer is variable region. Bifurcated region means you already know it is Y shape. So here also major part is constant region, only terminal part is variable region. Variable region, that means FAB ends, 2 FAB ends, variable region, fragment of antigen binding sites. So that's why variable region. Next question. The term humor refers to body fluid. Body fluid means plasma and lymph. Antibodies released by plasma cells in humor destroy antigens. That's why it is known as humoral immunity. Bone marrow, plasma and lymph, all internal tissues, all subcutaneous, only body fluids. And humoral immunity destroy only free antigens. Antigens not present in the cells. Circulating antigens. So answer is humor means body fluid. Body fluid means plasma and lymph. Next question. T lymphocytes first mature in, so that is thymus. Why they got the name T, thymus? The lymphocytes mature in bone marrow, B lymphocytes. The lymphocytes mature in thymus, T lymphocytes. In case of birds, the lymphocytes mature in bursa of Fabricius, known as B lymphocytes. Bursa of Fabricius. So it is a primary lymphoid organ, opens into cloaca in birds. So next question. An antigen presenting cell. First option, present antigen to T cells, correct. Secretes antibodies, wrong. Marks each human cell as belonging, so wrong. Belong to that particular person, so that is not correct. Secrete cytokines, wrong. Antigen presenting cells, mature B lymphocytes. Mature B lymphocytes. Macrophages. Macrophages means monocytes present in connective tissue. And dendritic cells. Dendritic cells, they are known as antigen presenting cells. Antigen presenting cell engulf the antigen, break the antigen into several antigen determinants and keep the antigens on the surface with the help of MHC2 molecule, major histocompatibility 2 molecules, on the surface of all antigen presenting cells. CD4 marker is present. Cluster differentiation 4, that is one type of protein, CD4 marker. While discussing HIV also we discuss. HIV, on the surface of HIV, glycoprotein 12041 are present. They recognize the cells contain CD4 marker. So antigen presenting cells contain CD4 marker. T helper cell also contains CD4 marker. So antigen presenting cell, so function of antigen presenting cell, they present the antigen to T cells, T lymphocytes. So that's why first one is correct. Now we discuss next question. Helper T cells stimulate B cells to mature and increase their antibody production. Helper T cells release T helper cells release interleukins. The interleukins stimulate the mature B lymphocytes. The mature B lymphocytes undergo division. Two types of cells are formed, plasma cells and memory cells. The antibodies produced by plasma cells involved in humoral immunity. So in this manner, helper T cells stimulate B cells to mature and increase their antibody production. So answer is C. T helper cells release interleukins. Interleukins stimulate mature B lymphocytes. Mature B lymphocytes undergo division. Two types of cells are formed, short-lived plasma cells, long-lived memory cells, B memory cells. The plasma cells produce and release circulating antibodies. The circulating antibodies produced by plasma cells involved in humoral immunity. So answer is C. Now we discuss next question. The secondary lymphoid system comprises of thymus and liver. Thymus is a primary lymphoid organ. Liver is a neither primary lymphoid organ nor secondary lymphoid organ. B. Spleen, lymphocytes, tonsils, mucosal lymphoid tissue are the secondary lymphoid organs. Tonsils are functional up to 8 years. Later they become non-functional. RBC, WBC and platelets, they are cellular barriers, not lymphoid organ. So answer is B. So next question. Memory cells are responsible for immunological memory, antigen production, or they are cells of cerebrum, cells responsible for memory in brain. So memory cells are not related to brain, they are related to immunological memory. So these cells have memory of the antigen. Whenever the same antigen enter into our body, the memory cells recognize the antigen and produce antibodies and plasma cells. So answer is responsible for immunological memory. Next one, recognition and digestion by the phagocytes due to the coated surface of antigens by the antibodies is known as opsonization, immunization, T cells immunization reaction, B cell immunization reaction, that is known as opsonization. What is opsonization? More susceptible to phagocytosis. So sometimes, so it is a monocytes. Now it is a cell contained antigen. The antibodies fail to destroy the cell contained antigens. Now the antibodies form a layer. Now the phagocytic cell quickly respond and destroy the cell-contained antigen. So in this manner, for example, the monocyte may destroy in 2 hours. Now it destroy within few minutes only. This process is known as opsonization. Opsonization means more susceptible to phagocytosis. The phagocytic cell always destroy the cell-contained antigen. So in presence of this coat, what happen? They quickly destroy due to coated surface of antigens by the antibodies. So it is the cell-contained antigen, or sometime it is the antigen antibodies attaches. So due to presence of antibodies around the antigen also, the phagocytic cell easily destroy the antigen. This process is known as opsonization. Next question. Which of the following immune response is more potent, more powerful and long-lived? Primary immune response only, secondary immune response only, tertiary immune response only, both secondary and tertiary. Whenever pathogen enter into organism or host body, the immune system respond, that is primary immune response. Primary immune response is slow. At the time memory cells are formed. If the same pathogen enter next time, the memory cells, you already know, memory cells produce antibodies, IgM quickly interact. So that is known as secondary immune response or anamnestic immune response. No tertiary. Third time enter also we call secondary. Fourth time also enter secondary only. So that is secondary immune response or anamnestic immune response. So answer is B. Next question. Active immunity is borrowed from an active disease case, developed in direct response to a disease agent, the product of borrowed antibodies. Passive immunity, that is activated. So you already know, active means antibodies are developed in the body, maybe naturally or artificially. During our day-to-day activity, we exposed to several pathogens. In childhood, every human suffering from chicken pox, at the time immunity developed. Due to this immunity, we never suffer from chicken pox. So in future, so that is active immunity. Similarly, vaccination also active. To prevent dreadful diseases, the parents inoculate, with the help of lab technician, inoculate the several dreadful pathogens cause diseases in killed form, live weak form or protein vaccination. So due to vaccination also antibodies are developed. You should remember, in our body antibodies are developed, that is active. What is passive? Antibodies not developed. So some dreadful pathogens enter. For the survival, we inoculate the preformed antibodies, that is passive. We develop antibodies in a sensitized organism, immunized organism. In case of snake venom, human body is non-immunized. Sheep, horse, immunized. We develop antibodies in sheep, horse, and simply we inoculate in our body. So answer is developed in direct response to a disease agent. Active immunity, natural active and artificial active. Passive means inoculation of preformed antibodies, already developed antibodies, that is passive. Passive also two types, natural passive, artificial passive. Mother without her notice transfer the antibodies to fetus via placenta, one example. Second, mother transfer the antibodies to infant through colostrum, that is another example. So next question. Which is the proper order of events in cell-mediated immunity. Antigen enter the tissue, so correct. Here also antigen enter. Macrophages engulf antigen. Antigen present to member. So whenever antigen enter, so lymphocytes should be stimulated. Lymphocytes should be stimulated. Lymphocytes should be stimulated. So lymphocytes stimulated. Next one is macrophages engulf antigen. T lymphocytes attack antigen bearing antigens. So that is correct. Macrophages engulf wrong. So answer is B. Cell mediated immunity. Antigens enter. Lymphocytes are stimulated. That is the T lymphocyte. Or we discuss cell mediated immunity. Antigen presenting cell present the antigens on the surface. Now T helper cells. What are T helper cells? T helper cells are lymphocytes. The lymphocytes should be sensitized, activated. The activated T lymphocytes release interleukins. The interleukins stimulate TC cell. The TC cell undergo division. TC memory cell. And effector CTL, that is cytotoxic T lymphocyte, also known as killer cell. Now the killer cell recognize the antigens present in the nucleated cell with the help of MHC1 molecule. So killer cell, TC cells contain CD8 marker, cluster differentiation marker. The CD8 marker recognize the antigens present on the surface with the help of MHC1 molecule. So now need antibody. With the help of antibody, the receptor of killer cell attaches to altered self cell. What is altered self cell? Due to presence of antigen, the function of the cell is changed, altered self cell. Later, killer cells release perforins. Perforins make hole in the cells. Later, killer cells release granzymes. The granzymes enter into the altered self cell through holes and stimulate the caspases. Caspases are the proteins. They stimulate the programmed cell death mechanism. As a result, altered self cell along with antigens destroyed. So this is the mechanism of cell mediated immunity. T lymphocytes should be stimulated. The stimulated T lymphocytes release interleukins. Interleukin stimulate TC cell. Antigen presenting cell submit the antigen to T helper cell. Antigen presenting cell contain MHC2 molecule. On the T helper cell, CD4 marker is present. CD4 marker identify the MHC2 molecule, that is one type of protein present on the surface of antigen presenting cell. Now T helper cells activated. Lymphocyte, T helper cell, one type of lymphocyte. Lymphocyte sensitization is important. That's why answer B. Now we discuss next question. Bursa fabriceus or fabrica is lymphoid organ in birds. That is primary lymphoid organ. The lymphocytes formed in bursa fabriceus are known as B lymphocytes. Next question. The most commonly transplanted tissue is cornea. Why cornea is the most commonly transplanted tissue, and it is easy also. No blood supply, no antigen antibody reactions. Antigen antibody reaction. You already know humoral immunity. So antibodies present in the body fluids, blood and lymph try to destroy the antigen. So no blood supply to cornea. That's why it is a common transplanted tissue is cornea, and this transplantation is easy when compared to transplantation of other organs. Next question, skin grafting within one organism within one organism is an example of. Within one organism means, okay, sometimes due to skin burn, for example, in one person skin is damaged, so near the wrist, at the time they take skin from thigh region, so they take the normal skin from thigh region. So this type of transplantation is known as autograft. What is autograft? Transplantation of skin or any organ tissue within the same person is known as autograft. What is allograft? Transplantation of organ between the organisms belong to different organisms of the same species. For example, in case of human being, the relatives, close relatives give liver, kidney, so cornea, so in this manner, so that is allograft. Transplantation of organ between the organisms belong to the same species known as allograft. What is isograft? The transplantation between identical twins known as isograft. No transplantation rejection reactions in case of identical twins. What are identical twins? The twins formed from single zygote. Identical twins. Identical twins are mirror images. They are morphologically, physiologically alike. Isograft. What is xenograft? Transplantation of organ between organisms belong to different species known as xenograft. Rarely nowadays they are using pig heart valves to replace the damaged heart valves in human. Pig belong to one species, human belong to another species. So transplantation of organ between the organisms belong to different species known as xenograft. So next question, name the chronic respiratory disorder caused mainly by cigarette smoking. Cigarette smoking means emphysema. What is asthma? You all know. Chronic disease, allergy. What is respiratory acidosis? The respiratory system not working properly, sufficient amount of oxygen not reach the organ. So carbon dioxide concentration increases, blood become more acidic. So that is respiratory acidosis. What is respiratory alkalosis? If we perform yoga, pranayamam, at the time we inhale we perform forceful inspiration. So due to deep breath, abundant amount of oxygen enter, oxygen concentration increases. So the blood become alkaline. So that is respiratory alkalosis. Decrease of oxygen, respiratory acidosis. Increase of oxygen in blood, respiratory alkalosis. Respiratory alkalosis is not a serious problem. We can rectify. Respiratory acidosis is a problem. In old people, lungs are not fully functional. So breathing, you already know tidal volume 500 ml. But in old people, the tidal volume is not 500, less than 500. So that is respiratory acidosis. So emphysema, you already know, alveolar wall damages, the damaged walls fuses, as a result respiratory membrane become thick. Even though abundant amount of oxygen present in alveoli, this oxygen failed to enter into pulmonary vein. So in this manner, in emphysema patient impair the pulmonary gases exchange. The carbon dioxide reach the pulmonary artery, fail to enter into alveoli. The oxygen present in the alveoli fail to enter into pulmonary vein. So emphysema, smoking. Next one, which one of the following is not a property of cancerous cell, whereas the remaining three are? Means a property of cancerous cell. They compete with normal cells for vital nutrients, correct only. They do not remain confined in the area of formation, means exhibit metastasis. They may exhibit metastasis due to absence of cadherin protein. In normal cell, cadherin protein is present, that's why they never separate. So due to absence of cadherin protein, they migrated to various organs, form the secondary tumors. They divide in an uncontrolled manner, correct. So what is cancer means? Uncontrolled cell cycle regulation. So failure of cell cycle regulation is known as cancer. So uncontrolled manner, correct. They show contact inhibition, wrong. Normal cells exhibit contact inhibition, means one cell, normal cell never touch the adjacent cell, contact inhibition. To maintain this property, normal cell never undergo rapid division. To maintain this property, normal cell never separate from the tissue. If any normal cell separated from the tissue, it may touch the adjacent cells. That's why the cancerous cells never maintained normal cell property. So fourth one is not the property of cancerous cell. So answer is D. Which of the statement is correct from the following statement? Gout is a kind of arthritis, correct. Accumulation of uric acid crystals in the joints, that is gout. Uricase enzyme is not synthesized. Uricase enzyme is not synthesized. It converts uric acid into urea. Gout is not a problem to kidney. Non-synthesis of an enzyme, uricase enzyme. Malignant tumor do not show metastasis, wrong. Malignant tumor exhibit metastasis. Benign tumor do not show metastasis. Diabetes mellitus, a communicable disease characterized by high blood sugar level. It is not a communicable, it is a non-infectious disease. So that's why wrong. Next one. 1st December is celebrated as World AIDS Day. So correct. So gout correct. Do not show wrong. So 2, 3 wrong. 1 and 4. So B option. Next question. HIV, the human immunodeficiency virus, belong to which of the following viral group? That is retrovirus. What is retrovirus? The virus participating in reverse transcription is known as retrovirus. What is reovirus? The virus present in respiratory system and intestine is known as reoviruses. Now COVID-19 also reovirus, respiratory system. What is rhabdoviruses? Rhabdoidae family. The virus belongs to Rhabdoidae family, rhabdovirus. So retrovirus means the virus participating in reverse transcription. So from single stranded RNA, double stranded DNA is formed. So retrovirus. All retroviruses cause cancer. So 33 answer is B. Next question. Azithromycin is a anti-AIDS drug. It delay the reverse transcription. It delay the reverse transcription. So prolong the lifespan. Anti-AIDS drug. Next question. Disease spread by droplet infection, common cold, smallpox, measles, all of these. So corona also disease spread by droplet infection. During sneezing, the droplets so comes out. So droplet infection or aerosols. in the form of aerosols. So all of these. Next question. Which disease is caused by the activation of proto-oncogenes? Cancer. Proto-oncogene converted into oncogene. So proto-oncogene undergo one mutation. Why? These are recessive alleles. For example, PP. So it undergo mutation, one capital P. So it unable to express its character. So proto-oncogenes undergo one mutation. If they undergo one mutation, they converted into oncogenes. Oncogenes stimulate the cell in which they are present. So the cell lost the cell cycle regulation, converted into cancerous cell. So answer is cancer. Shivering in malaria is due to toxin formed by breakdown of hemoglobin in RBC releasing. So how toxin is formed? Hemoglobin is a protein. Hemoglobin, in the hemoglobin, globin is a protein, hemo is iron part. This iron part converted into hemojoin. Hemojoin is brown color granule present in the cytoplasm of plasmodium. Whenever the shy joint rupture along with erythrocytic merozoites, hemojoin released into blood circulation cause the malarial symptoms: chill, shivering, cold, severe cold. So malaria fever relapse every 48 hours in 3 species except Plasmodium malariae. In Plasmodium malariae, erythrocytic cycle duration is 72 hours. That's why it causes quaternary malaria. Quarter means 4th day. The malaria fever is relapsed every 4th day in only one species of Plasmodium, that is Plasmodium malariae. In other species of Plasmodium, ovale, falciform, vivax, the malaria is relapsed every 3rd day. Every 3rd day means after 48 hours. Next question. Trypanosoma, the causative agent of African sleeping sickness, belong to which class of protozoa? That is Mastigophora. Mastigo means flagella. Trypanosoma contain flagella and it causes African sleeping sickness. First drowsiness, later sleep, so unconscious, coma, later death. African sleeping sickness. So intermediate host of trypanosoma, CC fly. Intermediate host. Man is the primary host. CC fly is a blood sucking insect. It acts as a intermediate host. CC fly. And trypanosoma contain flagella. That's why it belongs to class Mastigophora. Next one. Each cycle, erythrocytic schizogony in Plasmodium malaria is completed in 48 hours. Now only we discuss except Plasmodium malariae. Except in malaria, that is 72 hours. Malaria 72 hours. In other species 48 hours. So here Plasmodium malariae 72 hours. In the remaining 3 species 48 hours. So answer is C. Next question. The sporozoites that cause infection when a female Anopheles mosquito bites human being are found in salivary glands of mosquito. After formation in the female Anopheles mosquito, enter into hemocoel. From the oocyst enter into hemocoel. They, by performing locomotion, they enter into salivary glands. So salivary glands of mosquito. C. The continuous and excessive intake of alcohol causes or gastritis, problem in the stomach, fatty liver syndrome, liver enlarges, fail to perform its function, cirrhosis. So the liver cells are replaced by fibers, cirrhosis. If the person is suffering from cirrhosis, for the survival, only liver transplantation. So all of these. 31st May, no tobacco day. You all know World Health Day, April 7th. Cocaine is a crystalline alkaloid extracted from the leaves of, so cocaine, that is Erythrozyllum coca. Common names also important. Cocaine is known as coke or crack. Charas is a product of cannabis sativum. Cannabis sativum, flower tops, leaves and resin mixed in different combination. Charas, ganja, marijuana, hashish are prepared from cannabis sativum. So charas, it affects the cardiovascular system. So some sports persons also misuse, so artificially increase their stamina. Heroin is a diacetyl morphine is known as heroin. It is a opioid. Heroin is a depressant, slow down the body functions. What about morphine? Morphine acts as a painkiller. So diacetyl morphine, CH3COCl, diacetylation, addition of CH3COCl, acetylation. Diacetyl morphine is known as heroin.